It is important to understand the features of the most successful CAR T cells in patients with lymphoma, and with the goal to understand how to engineer CAR T cell therapies of the future to make clinical responses better. We found that through lineage tracing of CAR T cells across different time points, we found the specific cell subsets that expand from pre-manufacture apheresis to infusion products and beyond. And there we found that it is really the most quiescent T cells, the naive or central memory cells that expand, whereas affected T cells contract. However, a surprising finding for us was that T regulatory cells that are known immune suppressive subset of T cells also clonally expand from source T cells to CAR T cell infusion product. It is a really great instrument, and in our work we were able to do lineage tracing at scale in CAR T cells. And these findings that, like for example, the subsets that expand from pre-manufacture apheresis to infusion product allow us to design better manufacturing processes for CAR T cells in the future. So we have only the cell subsets of interest to growing out and not the cells we don't want. And in addition, just generally expands the biological understanding of CAR T cell therapies that are used right now.